Today, we're talking about the DNA strands and all the different missions it has to accomplish. Starting off with transcription, the way to make RNA to leave the nucleus. It starts off by opening up the DNA helix by the transcription factor binding to the promoter region with the RNA polymerase opening up the DNA double helix. Then it moves on to the elongation, which is the creation of RNA, which happens by the RNA polymerase building single-stranded RNA without a primer, and it's always made in a 5 to 3 direction. Such as, for example, would be our coding strand and this would be our template strand, creating our RNA using the template strand. Some important notes to remember is that the A binds to the U instead of the T, and the creation of RNA is continued until the termination sequence. So step 3, when it reaches the termination sequence, the nucleotides are added until the sequence, but since these nucleotides are A and T's, they're easy to break to the two hydrogen bonds. RNA polymerase comes off and stops transcribing. There's various termination methods, such as the hair loop method, and also the OR protein method for the RNA polymerase to stop transcribing. The post-transcription modifications include a 5-cap addition to the 5' end, which is an addition of 7 guanines. Also, a poly A tail, which is an addition of 50 to 250 adenine nucleotides. And lastly, then comes splicing. And splicing is to keep the coding regions to leave the nucleus while the non-coding regions to stay inside, which must be spliced out. So this is our intron, which must be spliced out in our RNA, and the spliceosomes come together to splice out the intron, which is a non-coding region. Mission accomplished. Our second mission is translation. So we start off with the mRNA, which enters the cytoplasm. The ribosome binds to the 5' cap on the mRNA and closes both its subunits on the mRNA. In the ribosome, there are various sites which can be a tRNA amino sites. So it starts off with the AUG binding to the P site, which enters with the codon AUG. Next, the amino acyl tRNA enters an A site, which creates a peptide bond with the P site, causing it to translocate to the E site where it exits. Also, the ribosome continues to translocate over a codon, which keeps repeating to create a protein formation of chain of amino acids. Such as in this diagram here, it starts with the P in the P site and continues on. An anticodon is a complementary codon which for RNA were located on the tRNA. Therefore, the RNA codes for the following amino acids. Due to stop codons, they release the chain from the ribosome. Mission